Imagine a medication originally developed to treat diabetes suddenly started doing something that even scientists never expected. Not just lowering blood sugar. Not just helping people lose weight. But something far more fundamental. Something researchers spent decades trying to achieve, and that now, unexpectedly, appears to be happening inside the bodies of millions of people around the world, without most of them even realizing it. This is the story of how a class of medications known as GLP-1 receptor agonists, the same drugs you know as Ozempic, Wegovy, and Mounjaro, began to spark one of the most fascinating conversations in modern science. And that conversation isn't just about weight loss. It's about human aging. It all began in the 1980s and 1990s, when researchers were studying a hormone naturally produced by the intestine after meals. It was called GLP-1, glucagon-like peptide 1. The work was highly technical, relatively obscure, and of interest mostly to diabetes specialists. The idea was simple: this hormone stimulated insulin release from the pancreas, lowered blood sugar, and signaled to the brain that the body had enough energy. If scientists could mimic this hormone artificially and extend its effects, perhaps they could create a more effective treatment for type 2 diabetes. After decades of research, the first medications capable of doing exactly that finally emerged. And they worked. They controlled blood sugar, reduced appetite, and produced significant, sustained weight loss, something no previous drug had achieved with such consistency. The world took notice. These medications became a global phenomenon. But then something began to appear at the edges of clinical studies. Something researchers had never planned to observe. The first clues came from major cardiovascular trials. The SELECT trial, published in the New England Journal of Medicine, followed more than 17,000 adults with overweight or obesity who already had established cardiovascular disease. Participants who received weekly semaglutide experienced a 20% reduction in the risk of heart attack, stroke, or cardiovascular death. That alone would have been headline news. But what surprised researchers was the speed and breadth of these benefits, and the fact that they didn't seem to depend entirely on weight loss. Even when scientists attempted to isolate the weight loss effect, many of the benefits remained. Something else was happening. And that's when the questions began to change. Why would a gut hormone be protecting the heart? Why would it be lowering blood pressure, improving blood vessel function, and reducing inflammatory markers in the bloodstream? The answer, at least in part, was hidden in the anatomy of something few people had considered. GLP-1 receptors, the molecular targets these medications activate, aren't found only in the pancreas and intestines. They're distributed throughout the body. In the heart. In the kidneys. In the liver. In immune cells. And critically, in the brain. That changes everything. Because when a medication activates receptors in all of these organs simultaneously, it's no longer treating just one condition. It's potentially interacting with fundamental biological systems that govern healthy aging itself. Think about what happens as we grow older. Low-grade chronic inflammation, that persistent state of immune activation that no longer serves to fight infection but instead slowly damages tissues over decades, gradually increases. Scientists call this process inflammaging, a combination of the words inflammation and aging. We've covered inflammaging in a previous episode. And this chronic inflammatory state has been linked to nearly every disease people fear after age 50, Alzheimer's disease, heart disease, diabetes, muscle loss, and cognitive decline. GLP-1 agonists appear to act directly on this inflammation. Across multiple studies, they have reduced inflammatory markers such as C-reactive protein, one of the most commonly used warning signals physicians monitor when assessing cardiovascular and neurological risk. And that brings us to what may be the most remarkable discovery of all. In June 2026, researchers at the University of California, San Diego published the results of a randomized, controlled clinical trial in Nature Communications, the gold standard of medical research. Roughly half of the 108 participants received weekly semaglutide injections, while the other half received a placebo. The researchers used a series of biological, epigenetic clocks to track cellular aging over 32 weeks of treatment. Epigenetic clocks are tools that detect chemical marks on DNA, changes that regulate how genes are turned on and off throughout life. These marks change in predictable ways over time, allowing scientists to measure not a person's chronological age, but their biological age. And that distinction is crucial. A 60-year-old can have the biology of a 50-year-old. Or the biology of a 70-year-old. What the researchers found was striking. Participants treated with semaglutide showed a broad pattern of slower biological aging across epigenetic clocks associated with inflammation, blood health, brain function, cardiovascular health, kidney function, liver health, and metabolic health. The medication slowed the pace of biological aging by approximately 9% according to the Dunedin Pace epigenetic clock. 9% may not sound dramatic. But in aging biology, it's enormous. The annual pace of epigenetic aging associated with mortality risk decreased by roughly 3 years in the semaglutide group compared with placebo. Not 3 months. 3 years of biological aging, in just 32 weeks of treatment. But there's an important detail that rarely gets mentioned when people discuss this discovery. The effects appeared across multiple systems simultaneously. The strongest signals were observed in epigenetic measures linked to inflammation, the brain, the heart, the blood, the kidneys, the liver, and metabolic health. That suggests the medication may be influencing aging-related biology across multiple organ systems at once. And that's important because aging doesn't occur in a single organ. It happens throughout the entire body. A medication that appears to slow these processes in parallel may be touching something much deeper than appetite regulation or blood sugar control. A complementary pilot study found that approximately 49% of participants treated with semaglutide experienced an increase in telomere length during treatment. Those same individuals also tended to walk faster, suggesting a potential improvement in physical function. Telomeres are the protective caps located at the ends of chromosomes. They gradually shorten with every cell division. When they become too short, cells stop dividing, a process that directly contributes to tissue aging. And then we arrive at the brain. And this is where the story becomes more complicated. For years, researchers noticed something intriguing in large population databases. People with type 2 diabetes who used GLP-1 medications appeared to develop dementia far less frequently than those using other treatments. In an analysis combining data from a Danish National Health Registry with cardiovascular clinical trials, people taking semaglutide or liraglutide had less than half the risk of developing dementia compared with individuals using neither medication. That observation led to the first major clinical trials designed to test whether semaglutide could treat established Alzheimer's disease. The EVOKE and EVOKE Plus trials, funded by Novo Nordisk and conducted with exceptional scientific rigor, evaluated the medication in patients with early-stage Alzheimer's disease. The results, presented at the CTAD conference in December 2025, perfectly illustrate where the science stands today. Semaglutide produced a significant reduction in high-sensitivity C-reactive protein, a key marker of inflammation, across both studies. Systemic inflammation decreased. The mechanism worked. But progression of established Alzheimer's disease was not significantly altered on the primary clinical endpoints. Current evidence suggests that semaglutide may have a stronger role in early prevention than in treating Alzheimer's once the disease is already established. And that, by itself, is an extraordinary scientific finding. It suggests there may be a window of opportunity, a period during which intervention can make a meaningful difference before neurological damage becomes irreversible. But at this point, we need to pause and be honest about what science knows, and what it still doesn't know. The studies on biological aging are promising, but they have been conducted in specific populations. We still don't know whether the same effects occur in people without HIV, without severe obesity, or across different age groups. The long-term effects, beyond 1 or 2 years, on biological aging still need to be evaluated in larger and longer studies. There are also legitimate concerns regarding muscle loss that may accompany the weight reduction caused by these medications, something particularly important for adults over 50. And the gastrointestinal side effects, nausea, vomiting, and digestive discomfort, are very real, causing a significant number of patients to discontinue treatment. Science is moving quickly. But it has not reached definitive conclusions. What we have today are signals. Consistent signals. Promising signals. And in some cases, surprisingly powerful signals. Signals that are directing billions of dollars in research and scientific attention toward a question that would have sounded like science fiction just a few years ago. So what do we do with all of this? Take a step back and consider the bigger picture. For decades, geriatric medicine viewed aging as an inevitability to be managed, a decline to be slowed through healthy habits, blood pressure medications, cholesterol-lowering drugs, exercise, sleep, and nutrition. All of those things remain essential. But what GLP-1 agonists are placing on the table is a different possibility. The possibility that we may be able to do more than treat individual diseases associated with aging. That we may be able to influence some of the biological mechanisms of aging itself. Chronic inflammation. Epigenetic deterioration. Telomere shortening. Insulin resistance. The processes that sit beneath so many age-related diseases. A large 2025 analysis published in JAMA Network Open followed more than 60,000 adults with obesity and type 2 diabetes. People taking semaglutide or tirzepatide showed significantly lower all-cause mortality and lower risks of stroke and dementia compared with patients using other diabetes medications. 60,000 people. Not a laboratory. The real world. And that brings us to the question scientists are beginning to ask out loud. Are these simply obesity medications with unexpected positive side effects? Or are we witnessing the first pharmaceutical agents capable of influencing some of the deepest mechanisms of human aging? The honest answer, for now, is simple. We don't know. And that uncertainty is not a weakness of science. It's science doing exactly what it is supposed to do, remaining rigorous, cautious, and endlessly fascinated by what it does not yet fully understand. What we do know is that GLP-1 receptors exist in the brain, the heart, the kidneys, and the immune system for an evolutionary reason. We know these medications are activating something within those networks. And we know that the emerging data are changing the conversation about what may be possible. Perhaps the most honest thing anyone can say today is this. For decades, scientists dreamed of finding a pill, or an injection, that could influence the most fundamental mechanisms of human aging. No one expected it to come from a gut hormone. And no one expected it to be injected into tens of millions of people before we fully understood everything it might be doing. Beyond the years. Because understanding how we age is the first step toward aging better.